Hello students, once again we are going to meet after the festival, Dasara festival. So belated wishes first of all, wish you happy Dasara. And now till we have started the biotechnology principle and process. Okay, just once again I'll do the brief revision of biotechnology, that is utilization of the biological organisms and their enzymes for the synthesis of the new products for the welfare of the mankind. And this biotechnology is used for the production of large yielding and also for the meat and the milk production in a larger amount to overcome the need of the population. Okay, and here two basic principles of biotechnology we are going to see, that is genetic engineering, and also we are going to see the maintenance of the sterile environment. So before going to that genetic engineering, we have to see what is exactly the genetic engineering is. So you can see the image here. Genetic engineering means doing some experiments with the genetical knowledge, and you should have the knowledge of the chromosomes, you should have the knowledge of the genes and all. So you can see the image here. A piece, if we want to improve the crop plant, okay, I'll show you the video also. If we want to improve the crop plant, or a large yielding, if you want to prepare that one, then what happens, the what to say uh, genes are extracted from one organism to the another organism and inserted to the another organism. That is genetic engineering. Now in your mind the question may be arise, how the gene is extracted from one organism, which enzyme is important for the extraction of the gene and all that. For your question, I'll give you the answer. But before that, just you watch this video of the genetic engineering. Then we'll go for the tools of genetic engineering. Okay. Now I'm going to play the video for you people. Okay. So the video is playing here. You can see the video. So they are
Doesn't
showing
this
the
term sound a
tomato.
little weird?
Tomato plant
You
body
might be
means
wondering,
potato
is it
plus
a tomato
tomato.
or a potato plant?
Hoping that you are
It's
seeing the
actually
video here.
both. Yes,
Tomato
you
means
heard me
what?
right.
Potato plus
In
tomato.
this
So you
single
can see
plant,
tomato
the
plant
tomatoes
body,
grow above the ground,
tomato
while the
upper side
potatoes
and the
grow
potato
below the
below
ground
side.
level.
That is
And
the
how is this made possible?
hybridization technique for
It can
the
be done by a
nowadays
process called
biotechnology is
grafting.
following.
In
So what
this,
is
the stems of
grafting?
both the
Also they are
tomato
showing.
and the
Grafting
potato
means,
plants
see, this is
are
the tomato
chosen
plant
and
body,
then joined
potato plant
together by
body.
tying
They
them.
are joining. The
This is
cytoplasm
an
of these
example of
plant
plant
bodies are joined,
engineering.
and
On the similar
then it is
lines,
called as
when we
plant
cut and
engineering
join
technique. So
genetic
you can
material,
see
the
the one
process is known
organism
as
is
genetic
gene
engineering.
genetic engineering. They are
It's
showing
one of the principles of
principles of biotechnology.
biotechnology.
So first one is genetic
Now let's
engineering.
explore more about these
So this is
principles in detail.
what they are showing, how the
Most of
gene
the
is
principles
extracted
of
and how the
biotechnology are
gene is
based on
inserted.
two engineering branches. That is, genetic engineering and chemical engineering. Genetic engineering is the branch which deals with the modification of the genetic material of an organism. And which genetic materials are we referring to?
So all these videos
We
you have
know
to
that
see
it's the
compulsorily.
DNA which is the genetic material for most of the organisms. And what about the remaining ones? Well, in them, it's the RNA which acts as the genetic material. Just like in the case of some viruses. So genetic engineering deals with the manipulation of genetic material. Moving ahead with the next principle, which is chemical engineering. The name itself suggests that the principle revolves around the use of chemicals for the growth and the maintenance of cell cultures. For this, the desired eukaryotic or microbial cell cultures are stored in a contamination-free zone. Further, these cell cultures are used for the production of vaccines, antibodies, enzymes, and various other useful products. We will learn about chemical engineering in the upcoming videos. For now, let's just discuss in detail about genetic engineering. Did you know that the first ever genetic modification was done in a bacterium? That's right. And later on, these modifications were applied to animal as well as plant cells. The genetic modification can be done either by adding something new to the existing genetic material, or it can also be done by deleting something from the existing genetic material. The DNA obtained after modification is known as recombinant DNA. Technically, genetic engineering can be done in many ways. Recombinant DNA is one way to pursue it. When genetic engineering is applied to modify organisms, they are known as genetically modified organisms, abbreviated as GMOs. And what organisms can be modified with this technique? Almost all living organisms can be modified with the help of genetic engineering. What kind of applications does this serve? Modifications in organisms can give rise to beneficial products. Consider plants, for example. With the help of genetic engineering, we can produce robust crops, pest-resistant varieties, and also high-yielding varieties of many crops. This was, in short, about one way in which genetic engineering can be carried out. The other way to perform genetic engineering is by making copies of the genetic material. This is also known as gene cloning. In simple words, gene cloning can be defined as the process of making an exact duplicate of the genetic material. But why do we need a duplicate of the genetic material? Let's consider an example to understand this concept. Imagine there's a new product launched by a company in the market. This has some really cool and amazing features which will be of great use to all. Now, is it fine if the company manufactures just one piece? Of course not. There will be so many people who will want to buy it. For this, there will be multiple copies of the same product manufactured on a large scale to suffice the demands. This just means,
Well, you have seen the video of the genetic engineering, that animation you have seen there. So in the animation, what they have shown is the DNA is cutted from one organism and it is inserted to the other organism, and then what happens there, the new offspring or the new DNA is going to organize or is going to synthesize, we can say, and that new DNA is called as recombinant DNA. The new DNA is called as what? Recombinant DNA. So the complete genome is changed over there. Okay, the complete genome is changed over there, and then we are going to see that this genome is producing, the changed genome is producing the its own offspring. Okay, so basically we are going to see the three steps in genetically modifying organisms. First of all, which is a step here, identification of the DNA with a desirable gene. Suppose if we want to insert any of the part of the DNA, then we have to identify first. Okay, so genes with a desirable genes means what? The suitable genes should be there in that sequence. Okay, so first step is identification of the DNA with a desirable gene. Second step is introduction of the identified DNA into the host, and the third step is maintenance of the introduced DNA in the host and transfer of the DNA to the progeny. These are the four steps. Actually, three basic steps what you have to follow to synthesize the genetically modified organism. So this is of 3 marks question. What are the basic steps in genetically modified organism? 3 marks question it is. Okay. Now we'll go for the next concept, that is tools of recombinant DNA technology. So in the previous class I have completed till here. Okay, that is up to genetically modifying organism. In the previous class I have completed till here. Now in the today's class I have to start the tools of recombinant DNA technology, students. So it is very important for 2 marks. Okay, 2 marks question it is. Mention the names of tools of recombinant DNA technology. Then here we can see that there are 4 tools of genetic engineering or recombinant DNA technology are there. First of all, we are going to see that the restriction enzymes we are going to see here. That is the first tool here. It is enzymes, polymerases enzymes and ligases. Second one is vector, and third one is actually bioreactor should be there. Okay, the third tool of genetic engineering is bioreactor. They have not mentioned. and the fourth one is host organism. Okay, the fourth one is host organisms. Write down the four tools here. The four tools of genetic engineering are, first one is enzyme. In the bracket you have to write the restriction endonuclease enzyme and ligase enzyme and polymerase enzyme. These three enzymes you have to write in the bracket. First one is enzyme. Second one is vectors we are going to see. And the third one is bioreactor, where it is not there in this, it is not there, bioreactor. And the fourth one is host organism. Fourth one is what? Host organisms. So here 2 marks question it is, mention the 4 tools of genetic engineering. Okay, mention the 4 tools of genetic engineering. Well, next we are going to see, the first we will start the enzymes. The students will say continuously that DNA is extracted from one organism, inserted into another organism. How it is possible? See, DNA is present in the complete inner built, it is inner built we can say. DNA is a molecular basis, it is inbuilt. How it is possible to identify the DNA? That is, first of all, it is possible by the enzymes we can say. So enzymes are very much important in the case of biotechnology. They play a vital role. They play an important role in the field of biotechnology. Why? Because without these enzymes, there is no extraction of the DNA from one organism and the DNA is not joined with each other. That is why we will say that the enzymes plays a very important role in the field of biotechnology. Now, how many types of enzymes are there? So, basically, in the biotechnology, there is a presence of 2 types of enzymes. In the biotechnology, how many types of enzymes we are using? 2 types of enzymes we are using. First one is called as restriction endonuclease enzyme. Which is the first enzyme here? Restriction endonuclease enzyme. And the second enzyme is ligase. Which is the second enzyme? Ligase here. Okay. So, what about the restriction endonuclease enzyme? Restriction endonucleases are also called as molecular scissors. What they are also called? They are also called as what? Molecular scissors, or they are also called as molecular knives. What do we say here? They are also called as molecular knives. Knives. Molecular knives. So here, these molecular scissors or molecular knives, who has organized this? That is, who has discovered these enzymes? That is, by the scientist called as Hamilton Smith. Hamilton Smith is the name of the scientist who has discovered the restriction endonucleases enzyme, where he has discovered in the bacteriophage, okay, in the bacteriophage and also in the E. coli, okay, in the bacteriophage and also in the E. coli. And first restriction endonuclease enzyme is HIND2. HIND2 is the first restriction endonuclease enzyme which has been identified by the scientist called as Hamilton Smith from the bacteria called as Haemophilus influenzae. What do we call? Haemophilus influenzae. Okay. So, I'll repeat it once again. So, how many tools of genetic engineering are there? 4 tools of genetic engineering are there. That is, the first one is called as enzyme, second one is called as vector, third one is the bioreactor, fourth one is the host organism. Now, we are going to see about the enzymes. So, what are enzymes here? These are the catalysts we can say. These are the biocatalysts where they are cutting the DNA at the specific site and also they are joining the 2 DNA fragments. These are the enzymes. So how many types of enzymes are there here? That is, two types of enzymes are there. One is called as restriction endonuclease enzyme. Other one is called as ligases enzyme. One is called as what? Restriction endonucleases enzyme. Other one is called as ligases enzyme. So this restriction endonucleases and ligases are those enzymes which first of all we have to see what are restriction endonucleases. So restriction endonucleases are those enzymes which are cutting the DNA at the specific site. What they are doing? They are cutting the DNA at the specific site. That is called as what? Restriction endonuclease enzyme. So, they are also called as molecular scissors, or they are also called as molecular knives. Knives, knives. Molecular scissors or knives, and these molecular scissors and molecular knives, or we can say molecular knives, has been extracted by the scientist called as Hamilton Smith. So students, Hamilton Smith overall, he has just identified the 900 restriction endonuclease enzymes from the 230 bacterial strains. Okay, from the 230 bacterial strains. Understood? So these restriction endonucleases enzymes were first identified by the scientist called as Hamilton Smith, and also here what happens, these enzymes are extracted from the bacteria called as Haemophilus influenzae. That spelling is not there here. It is extracted from the bacteria called as Haemophilus influenzae. Okay. And here students, this Haemophilus influenzae is the name of the bacteria. But the name of the endonuclease enzyme is HIND2. HIND. HIND, Roman number 2. This is the enzyme you can see on the screen. This is the enzyme which has been extracted from the bacteria called as, that is, Haemophilus influenzae. Okay. And also these restriction endonucleases can also be seen in the bacteriophage as well as E. coli. Okay, I'll show you the picture, don't worry about this, but you have to keep the point in your mind that REN means restriction endonucleases. Define what is REN, expand REN, restriction endonucleases, 1 mark question. Then which enzyme has been discovered first as an REN, that is HIND2. HIND2 has been extracted from Haemophilus influenzae. How many restriction endonucleases are discovered? 900. From how many strains? 230 strains. Like this. Like this, the restriction endonucleases has been synthesized. Okay. So 900 restriction enzymes are there. More than 200 to 30 strains of bacteria. Well, this is about the introduction of the restriction endonuclease enzyme. Now, how the restriction endonuclease enzyme is named, that we have to see. So, how it is named? First of all, the first letter of the name is coming from the gene. Okay, the first letter of the name is coming from where? It is coming from the gene. Now, students, gene, for example, here we have to take a bacteria. Okay, here we have to take what? Here we have to take bacteria. So that bacteria is a prokaryotic organism, and it is grown. It is a prokaryotic organism. That means indefinite nuclear membrane, we will say. Prokaryotic cell organism which has been isolated in the laboratory condition, called as Escherichia coli RY13. Okay, called as what? Escherichia coli RY13. So, in short, it is named as what? EcoR1. Okay, in short, it is named as EcoR1. So, first letter of the name should be always generic. Instead of writing comes from the genes, you should write the first letter should always generic. That you have to write. Then the second letter, the second two letters from the species of the prokaryotic cell from which they are isolated. Okay. So second step says that since they are prokaryotic organisms, they can be admitted, isolated and admitted in a particular organism. Okay, for example, E. cor1 is a uh, name of the bacteria. So E. cor1 is it is the name of, it is a enzyme extracted from the E. coli bacteria. Okay, E. cor1 is the enzyme extracted from the E. coli bacteria. That is the second step. Always you have to remember that the second letter should be specific. The first letter should be generic. The second letter should be specific. Next letter should be should have the number of strains like Roman number 1, Roman number 2, Roman number 3, like that number of strains will be there in the next letter. Then fourth step is the Roman numbers following the names indicate the order in which the enzymes are isolated from the strain of bacteria. See, for example, E. coli bacteria is there. The E. coli bacteria alone is giving the 25 enzymes. Then how can you name is EcoL1, EcoL2, EcoL3, EcoL4 up to EcoL25. Like that the Roman numbers will be indicated for each specific strain of the bacteria. Okay. So once again I'll repeat. The first rule of naming the biotechnological restriction endonucleases enzyme is it should be in the capital letter. The first rule is it should be in the capital letter. Always the first letter should be in the capital letter. Then the second point says that it is generic, that means specific we can say. The second two letters of the species will be indicating as specific. Then the third one is next is derived from the number of strain. Then the fourth one is number of Roman numbers following the names indicate the order in which the enzymes were isolated from that of bacteria. That means the Roman numbers are given to these bacteria so that if we go wrong also we can easily identify that Roman number, and then we have to do the work with the experiments. Okay, this is about the naming of the enzymes. Next, well, we are going to see action of enzymes. So restriction enzymes belong to a larger class of enzyme called as nucleases. Students, restriction endonucleases enzyme. Restriction endonucleases enzyme has been extracted from the nucleases products. Has been extracted from the nucleases products. So, these are two kinds of nucleases. One is called as exonuclease and the other one is called as endonuclease. Is it visible for you? That is exonuclease and the endonuclease. Further, exonucleases remove nucleotides from the DNA, whereas the endonucleases makes cuts at specific positions within the DNA. Well, here you can see exonuclease, what it will do, it removes the nucleotide. It removes the nucleotide from the DNA sequence. What the endonuclease will do, it will cut the DNA at the specific site where the two sugar phosphate bonds are binded. That is called as DNA. Okay, that is called as what? Restriction endonuclease enzyme. Okay, once each restriction endonuclease function by this inspecting the length of the DNA sequence. Once the DNA and cut each of the two strands of the double helix at specific point in their sugar phosphate backbone. Overall, students, overall conclusion is restriction endonuclease enzyme has been discovered by the scientist called as Hamilton Smith, which and here he has extracted from the bacteria called as Haemophilus bacteriae, and he has after that he has extracted 900 strains of bacteria and 230 strains of storage. And here we are going to see from the 230 strains of the bacteria, there is an extraction of 900 restriction endonucleases, and all the 900 restriction endonucleases will cut the DNA at the specific site. Okay, and that specific site is called as palindromic nucleotide sequence. What do we call for that? It is the palindromic. It is the palindromic nucleated yes. Have you understood? So what restriction endonuclease is done here? So restriction endonuclease is cutting the DNA at the specific site, and then it is called as palindromic sequence. Okay, that specific site is called as what? Palindromic sequence. Now till now, hope so you have understood. Once again I'll repeat, please concentrate here. In the today's class, what we have seen. So in the today's class, we people have seen the tools of recombinant DNA technology. So how many tools are there? 4 tools are there. That is, first of all, the enzymes. Second one is vector. Third one is a host organism. Fourth one is bioreactor. This is very important for 2 marks. Name the tools of genetic engineering, or they will ask name the tools of recombinant DNA technology. Next we can see restriction endonucleases enzyme. What are these? REN, restriction endonucleases enzyme. First point. Second point is they are also called as molecular scissors or molecular knives. Second point. Then the third point is they cut the DNA at the specific site. What they will do? They cut the DNA at the specific site. Then fourth point is there are this REN has been discovered by the scientist called as Hamilton Smith. REN has been discovered by the scientist called as Hamilton Smith. Where he has discovered? In the bacteria called as Haemophilus influenzae, called as HIND2. There we have seen the discovery of the REN. Then how many restriction endonucleases are there? 900. From how many strains? 230 strains of bacteria. Which is the first one? That is HIND2. HIND2 is nothing but Haemophilus influenzae bacteria is responsible for the enzyme called as HIND2. Okay, HIND2 is nothing but it is a sequence of 6 base pairs. Complementary sequence, what do we say? Complementary base pair, what do we say? That is called as what? The sequence. Okay, so HIND2 is the enzyme which has been extracted from the bacteria called as Haemophilus influenzae. Next you can see how the restriction endonucleases are named here. Naming of an enzyme, restriction endonuclease enzyme. So first letter always it should be generic name. See, write the points. First letter is generic name. Second letter is the specific name, species name. What is the second letter? It should be the species name. Then, if one bacteria is extracting nearly 14 to 25 strains, then sequentially the Roman number is given. Next letter is derived after the specific name or the after species name, the insertion of the Roman number is done. Okay, Roman numbers are indicated in which order in which the enzymes are just they are kept and they have kept and how they are working there. Okay, that they are shown in the restriction endonucleases enzyme. These are the namings. Okay. Then this restriction endonuclease enzymes are of 2 types. Okay. So first of all, students, naming of the enzyme is asked for 3 marks. Okay. Write the name rules of naming the enzyme REN, that is for 3 marks. Then this introduction of the REN is for 2 marks. That is how the REN is synthesized, who has discovered this one, how many are there, which is the first one, first REN. All those are involved in the 1 mark question, and also even complete introduction is involved in the 3 marks. Okay, this one is a 3 marks question. 10. Now coming to the 2 marks, once again 3 marks question, I'm sorry. Once again 3 marks question. How many types of RNA nucleases are there? 2 types of nucleases. That is restriction endonucleases and exonucleases. Exonucleases and endonucleases are the 2 types of nucleases. What is the function of the exonuclease? Exonuclease removes nucleotides from the end of the DNA. That is, when the DNA is cutted, it is removing the nucleotides. Whereas endonucleases makes the DNA to cut the specific position. Okay, endonucleases is cutting the DNA at the specific position. Okay, that is the work of the endonuclease. After that, this endonuclease is responsible for the DNA sequence. Okay, endonuclease is responsible for the DNA sequence. That is, we can say about the action of the enzymes here. So here once again 2 marks question will be there. Name the 2 types of nucleases which is present in the REN, that is exonuclease and the endonuclease. Once again define exonuclease, that is 1 mark. That is it is removing the nucleotides from the ends of the DNA, whereas define exonuclease or endonuclease, if it is asked for 1 marks, then cut the DNA at the specific site. That is the 1 mark question and 2 marks question. Okay. Next you see, this is called as palindromic sequence. Students, what is the meaning of palindromic sequence? Write down the definition. It is a sequence where the 6 nitrogen base pairs are similar to read in both forward direction as well as backward direction. Okay. Restriction endonucleases enzymes are those enzymes which have needed always in the back, which are readed in the always readed in the backward direction as well as forward direction. That is called as palindromic sequence. For example, you can say madam, M-A-D-A-M. If you read from the front side, it is madam. If you read from the back side, it is madam. Okay. So wherever you go, whichever you go, that is different matter. But here the what to say, the 6 nitrogen base pair without changing its sequence, maintaining the complementary base pair, that is called as what? Palindromic sequence. What do we call for that? Palindromic sequence. Okay. Now, here students, this palindromic sequence is also asked for 1 mark question. Define palindromic sequence. So here you can say that it is the sequence where there is a presence of 6 nitrogen base pairs which is reading similar in both forward and the backward direction. Okay, that is called as what? Palindromic sequence. Well, here we are going to see the one more enzyme name, that is ligase. So this ligase enzyme and the vector class we will see in the next session. Okay, thank you students.